Hey guys, so today we're getting into the details of DNA structure. So, whoops, there we go. Alright, let's have some quick things that we're going to be looking at in this video. What does DNA stand for? Of course, that'll lead us to our structure. What is DNA? What is the structure of DNA? And of course, we're going to talk a little bit about DNA replication as well. So, we haven't already guessed or already heard or learned, DNA stands for deoxyribonucleic acid. And DNA is our genetic code. Sometimes you might say it makes us who we are. We'll get into the details of what that exactly means in just a little bit. So our DNA alphabet, how many nucleotide bases in DNA are there? Well, the answer to that is 4. And you know those bases. The names of them are guanine, adenine, cytosine, and thymine, or we can abbreviate them as G, A, C, and T. So those are the 4 main bases that you will need to know. Now, we've talked about genetics a little bit already, and we know that genes contain instructions to make proteins. So, the simple version of this, or the simplified version of this, is that we start with our genotype, or the information that's stored in the DNA. There's an RNA copy of it made, and then that produces a protein, which will then translate into our phenotype. So we'll go over this in much more detail when we get to actually protein synthesis. But we know that proteins are made of amino acids. Those amino acids come from codons that are in our mRNA, and then that RNA is going to come from the DNA, which is in the nucleus of our eukaryotic cells. So in the human genome, we have how many DNA bases? Take a quick guess. And the answer is 3 billion. So that's what this number looks like. It's quite a bit, quite a big number. And so we're not all exactly the same. What percent of your DNA do you think is similar to other people in the world? So take a quick guess. It's actually 99.9%. Now that seems like a large number that's similar to somebody else, but when you actually do the math, that's still 3 million bases that are different from your DNA to any other human being on Earth. So that's quite a few. We'll talk about the implications of this when we get to DNA fingerprinting and forensics and evolutionary relationships with DNA. Let's get back to our basics first. So today we're first going to talk about DNA structure, and then we're going to move into a brief review of DNA replication. So we've already gone over the history of the discovery of DNA, and remember there were lots of scientists that contributed to our final structure as we know it now. Of course, Watson and Crick get the credit. They got the Nobel Prize along with Wilkins, who worked in a lab with Rosalind Franklin, who did not deserve a lot of the credit for a very long time, but her photograph taken as an X-ray diffraction image was really, really essential to Watson and Crick's work and how they were able to conclude the structure of DNA as we know it now. So we know it's a double helix. So for a long time, a lot of scientists argued over what kind of structure the DNA would be in. Tetranucleotides, which is what this is right here, was one of the hypotheses. But at least now we have come to the experimental evidence, and we have reasoned that DNA is a double helix with our base pairs on the inside and our sugar phosphate backbone on the outside. We know now that there's about 10 base pairs per turn in our double helix, and DNA is very, very small, but we can cram a lot of it into one nucleus. Alright, so let's talk about our nucleotides. Remember, these are our monomers of our large nucleic acids. So our nucleotides are made of 3 main parts: our 5-carbon sugar, which in this case is deoxyribose, a phosphate group, which is here, and then of course a nitrogenous base, which is either our A, T, G, or C. Now the sugar and the phosphate are going to make up the backbone of the DNA, and we call that we call the sides of the ladder, and of course the A's, T, G's, and C paired in the middle are going to make up the rungs of our ladder. So this is why we call DNA a twisted ladder. Alright, again, so looking at the structure of nucleotides really quickly, again, the phosphate is always generally represented as a little circle on the end. It's attached to the 5-carbon sugar, and then, of course, our base could be either A, T, G, or C, so feel free to take a drawing of that in your notes. Alright, so when we look at DNA, we put it all together. We say that DNA is anti-parallel because on this end of our backbone we have our 5' sugars, our 5' carbons of our deoxyribose sugar, and down here we have our 3' carbon of our deoxyribose sugar. But on the opposite end, we're going to have the opposite direction. So the 3' will be up here and the 5' will be out here. As we elongate DNA, and we'll talk about this when we get to DNA replication, replication always goes from 5 to 3. You can see here that the A's, T's, G's, and C's are connected by hydrogen bonds in the center, which are strong enough to hold the DNA together, but not strong enough that they cannot be taken apart in circumstances where we need it to be split apart, such as in DNA replication or protein synthesis. Okay, so remember your base pairing rules. A is always pairs with T, and C is always going to pair with G. But our C's and T's are structured a little bit differently from our A's and G's. Now, our A's and G's are a type of nitrogenous base we call purines, and these are actually 2 carbon-nitrogen rings put together. Or pyridamines are just 1 carbon-nitrogen ring put together, and each of these has its own special molecular structure. Now, A will pair with T, and they will form 2 hydrogen bonds between each other, and G will pair with C, and they will form 2 hydrogen bonds. Now, it's not unheard of to have different or mistakes within the base pairing, and those will, of course, lead to mutations. But generally, the way that the molecular structure works, the A's and T's are more likely to pair with each other than the C's and the G's. All right, so here we are again. Take a quick sketch of DNA in your notes if you feel like you need to do so. Our phosphate deoxyribose backbone, of course, is on the outside, and we're going to take one more look at this sugar really quick. So as we see, this is a 5-carbon sugar. Of course, there's an oxygen up here as well. The 1' is here, the 2' is here, 3' is here, and this is at the bottom end of our DNA, or our growing end. The 5', of course, is going to be at this top. So the 5' will be sort of at the existing top of our DNA strand. Now, you don't need to memorize the structure of this sugar, just know that the 5' end is indicating the 5' carbon within our deoxyribose sugar. Alright, so how do we get all of our DNA packed into one small nucleus? Now, this is the video that I showed you guys just recently created with every single chromosome accounted for in the human nucleus. So how do we get all of those in there? And of course, the DNA is very, very long. If we stretched it all out from one cell, it would be about 6 to 5 feet, which is a huge amount of DNA. So we have different levels of packaging in eukaryotic cells. Now, of course, our DNA double helix is only 2 nanometers long, but of course, it's much longer when we stretch it all out. So what happens is we are going to have to wrap this DNA around what we call histones. Now, a histone is a special type of molecule, and a nucleosome is going to be 8 histone molecules put together. And so this DNA will wrap around the histones to create a nucleosome. And then that turns into 11 nanometers, and then the histones will pack together. And of course, this is how it looks as it's condensed into a chromosome. And the chromosomes are the parts of the DNA that we can see when it's condensed going through cellular division. So taking a look here again, we have our nucleosome beads, which is, again, 8 histone molecules. And wrapped around one of these nucleosomes is going to be about 146 base pairs. Now, again, this is only about 10 nanometers long. But each of these different histone complexes will be important for our packaging of DNA. Now, this is the quote that we've talked about from the Watson and Crick paper. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So before they were even done publishing their paper about the structure of DNA, they were already thinking about how could this DNA self-replicate, or how could it be copied into other cells within the same organism, knowing that we have the same DNA in all of the cells of one particular organism. So looking at that, DNA is structured very well to copy itself. Now, the main things we're going to need to remember about DNA replication are it happens in the nucleus. It happens during S phase of our cell cycle. Remember, S stands for synthesis. This is going to happen between G1 and G2. DNA is semi-conservative. We'll talk about, or DNA replication is semi-conservative, and we'll talk about what that means in a little bit. DNA replication occurs in the 5' to 3' direction. Excuse me. And the Messelson and Stahl experiments confirmed this semi-conservative replication structure. All right, so let's take a look at the different ideas scientists had before they were able to conclude that DNA replication was semi-conservative. Now, some scientists thought it was just conservative, meaning we had one strand of DNA. Remember, it's a double helix, so there's 2 strands actually put together. And then the original DNA was left over here, and then we would have completely new over here. Semi-conservative was the idea that when DNA was replicated, we'd have a template strand here, and then a new strand would come up here, and then we would have, each in our 2 new DNA molecules as a result in replication would have 1 old and 1 new strand. Now, this new meaning it's just coming from free-floating nucleotides within the cell, old meaning it came from the original molecule. And then the dispersive hypothesis thought that the DNA got a little bit more mixed up, and there were different segments of it in different sections when replication, of course, occurred. So when Messelson and Stahl did their experiments, they were able to conclude it was semi-conservative by marking some of the DNA with this heavy nitrogen isotope. So they used a N15 or nitrogen 15 isotope, and then they could measure the DNA and where it appeared and which was originally coming from the original strand and what was new based on their experiments, which is pretty cool. All right, so let's talk briefly about how replication works. So when we have a DNA molecule, remember this is all happening in the nucleus, special helicases, now these are enzymes, remember they end in ASC, are going to unwind the parental double helix. Then these single-strand binding proteins, we're going to stabilize the unwound parental DNA. So these proteins are important to make sure that the DNA doesn't reattach. Then the leading strand, which is going to be the one that is replicated in the 5' to 3' direction, is going to be synthesized continuously by DNA polymerase. And this is different from our lagging strand because that'll be synthesized discontinuously. So the lagging strand is the other end. The lagging strand is synthesized discontinuously. And primase is going to synthesize a short RNA primer, which is then extended by DNA polymerase to form an Okazaki fragment. And these are just these fragments that are going to be synthesized little bits at a time until DNA ligase is going to come through. So after the RNA primer is replaced by DNA, so it's another DNA polymerase, but it's not shown in this diagram, DNA ligase is going to join the Okazaki fragments together of the growing strand. And then, of course, our overall direction of replication is going to be this way from 5 to 3. And these are synthesized 5 to 3 as well, but they're going to start up here. So we'll go 5 to 3, 5 to 3, 5 to 3, 5 to 3. And that is how our 2 new DNA molecules will be grown. And this will continuously unwind the double helix until we have 2 completely new daughter strands. But of course they're not completely new because the replication is semi-conservative. So one side of the new strand will be from the original, and then one side of the new strand will be from new free-floating nucleotides within the cell. All right, so here, I have a quick question here, and I want to make sure you guys can see it all, so I'll just read it to you. It's just a really quick practice question. You should be able to answer it in a jiffy. When DNA replicates, each strand of the original DNA molecule is used as a template for the synthesis of a second, complementary strand, which of the following figures most accurately illustrates an enzyme-mediated synthesis of new DNA at a replication fork. So let's pull up the image. There it is. Okay, so if we look at A, B, C, and D, take a quick second to pause the video, try to figure it out. Okay, and our correct answer is D. So right here we see that the 3' down here, this is going to be part of our template for our leading strand because it'll be synthesized from 5 to 3. Remember, DNA is anti-parallel, so if it's 3' here, it'd be 5' on the other side. So 5 to 3, it'll grow in this direction. And then, of course, our Okazaki fragments on the other side will still be synthesized from 5 to 3, but in different fragments or segments. Okay, so that's it for DNA structure and replication. Be looking forward to the next video.